Good Monday morning, everyone. Welcome to week 3 in Bio 120. Let's see what we've got going on this week. So last week we studied about the chemistry of life, the importance of water in cells. It absorbs heat, it's used as a solvent, allows movement of things around inside the cell. The fact that carbon is the backbone of all the organic molecules because it can form such complex structures. And you learn some about the organic molecules like carbohydrates, lipids, proteins, and nucleic acids. And so hopefully now you have a background and just a little bit of chemistry to help us as we go through discussions for the rest of the term. The topic this week is all about classification, types of organisms, and their structures, the cellular structures. Classification is important because it helps us predict behaviors, predict what you will find in a particular organism if you know a classification. The highest level of classification is the domains, and there are 3 domains. Eukarya, which means true nucleus, the eubacteria, which are the true bacteria, and the archaebacteria, or the domain archaea, which contains bacteria that are found in very extreme environments, like extremely salty bodies of water, down in the depths of the ocean, in the hottest bodies of water on the planet, like hot springs, even boiling water, you'll find bacteria. And these are all archaebacteria. So 2 of the domains contain bacteria, the eubacteria and the archaea. And then that 3rd domain contains the eukarya, or eukaryotes is what we say. And eukaryotes are different from bacteria. So we got bacteria and the eukaryotes. Bacteria are also called prokaryotes. Okay, prokaryote and bacteria, those are interchangeable terms. The eukaryotes include plants, animals, fungi, and protists. And protists are single cellular organisms to multicellular organisms that cannot be grouped in any other kingdom besides plants, animals, and fungi. But they're still eukaryotes. And they're like an amoeba, paramecium, the algae, etc. Those are protists. Okay, so classification is very important, and one thing that you need to learn right away is the difference between prokaryotes and eukaryotes, the bacteria and all the rest of the organisms on the planet. Prokaryotes have very simple cellular structures. So they have cell walls, almost all of them do. They have a membrane, which separates the inside of this organism from the outside environment. And then they have some molecules on the inside of their cells, like DNA. DNA is not found in a nucleus in a bacterial cell. On the other hand, we have the eukaryotes that have very complex structures, including what are called organelles. So organelles are little compartments inside cells, like a nucleus, like a mitochondrion, like a chloroplast, like the Golgi apparatus, the endoplasmic reticulum, and there are lots more. So eukaryotes have complex cells, prokaryotes have simple cells. They have very different structures, and this is important to us. We'll see in just a minute. I'll mention this again in a minute when I get to another section. So what do you need to read? I have your chapters listed here. So chapter 20, section 1. That's all of that chapter that you need to read. And then chapter 4 in your OpenStax textbook. Please do read through this information. This is where you're getting your information for this course. So I see a lot of you who are using other sources. That's okay as backup, but you need to read through these sections in this textbook in order to get the correct information. Okay? So do that. Read the Module 3 overview and watch the videos. I also will have a video posted in this announcement about a tremendously interesting study that was done by Harvard Medical School, I believe, about antibiotic resistance in bacteria. It's only an 11-day study, and it shows you how fast bacteria become resistant to antibiotics when they are exposed to these antibiotics. Watch this video. It is just tremendously impressive. Great study, and it just shows you how fast this process happens. Okay, then what do you have to do this week? You have discussion board postings. You're going to have these every week except the last week. So you have something due by Thursday, your initial post, and then you have 2 response posts due by Sunday evening. So you're going to talk about why antibiotics, which are drugs that we take or put on our bodies to help with bacterial infections. Why do antibiotics target bacterial cells and not ours? So this is a very good question because this is a chemical that you're taking into your body to kill cells. But what you're hoping is that the bacterial cells will die and your cells will not. So why is it that these chemicals kill the bacterial cells and not ours? So here's where the difference in the cell structures comes up again. Because bacterial cells are so different from ours, these antibiotics target some of those particular cell structures that bacteria have that we do not. Okay, so this will be important when you look up this information. You're also going to discuss how our antibiotic use, how we use antibiotics, how much we take them, what we take them for, who we give them to, how the use of antibiotics increases the rate of antibiotic resistance in the bacteria. An example of this is a superbug, we'll call it a superbug, MRSA, methicillin-resistant Staphylococcus aureus. Of course, I'm a microbiologist, so I love all this kind of stuff. But MRSA has been around for a while now, a couple of decades. And it is not only resistant to methicillin, it started out being resistant to penicillin. So Staph aureus right now is resistant to penicillin. You cannot take it or be prescribed penicillin for a staph infection because it won't kill these bacteria. All right, then a whole other group of antibiotics, which we just group together and call methicillin, MRSA became resistant to, and unfortunately now we're also seeing a third tier of antibiotic resistance called vancomycin-resistant Staph aureus. And vancomycin is given in hospital because you get it intravenously. And so some cases of MRSA are developing greater resistance even than what they currently have, and so they are tremendously difficult to treat. The problem with this is that we are running out of antibiotics to use for bacterial infections. And this is true in a number of different types of infections. Some of the STDs are extremely resistant. Some of the bugs that you pick up in a hospital, like Pseudomonas aeruginosa, I got an infection from that after a surgery, very resistant to antibiotics. MRSA, tuberculosis, they're all different examples of this, and we're getting to the end of our arsenal of antibiotics in order to treat these infections. How are we going to combat this? Develop new antibiotics as quickly as we can, but if you're a pharmaceutical company, it doesn't pay for you to do a lot of research about antibiotics, because how long do you take an antibiotic? A week, 10 days, and then you're finished with it. A pharmaceutical company would much rather develop a new high blood pressure medicine or a different treatment for someone with diabetes or some other type of heart disease, because you're going to take that for the rest of your life instead of a week to 10 days. So there are financial pressures here that keep new developments from occurring in antibiotic research. The government is funding a lot of this now, which will help us all. So anyway, that you'll be discussing in the discussion board postings. You're going to begin your final project this week. So milestone 1 is due this week. If you don't see a topic on that list of topics, which I'm going to bring up in just a second, that you like, you need to email me to get an okay to do some other topic. And I explained all this in my announcement last week. So if you're not sure about it, go back and listen to the end of that announcement video for last week, and I talk about every one of these topics. Okay, and yes, my face is covered up, but that's all right. Here's the list. I took this right directly off your Milestone 1 guidelines and rubric. That is it. Here are the different topics: biodiversity, human population, air pollution, water pollution, whatever pollution that you can think of, waste management, etc. And I want you to notice down at the bottom, here are the elements that you need to include in this Milestone 1. An introduction. Notice that this is written in outline format. You will produce your Milestone 1 in outline format. This is not a journal entry. This is not an essay. And I cannot stress that enough. Excuse me, this is not an essay. So produce this in outline format. So topic 1, introduction, write that out. And then give me a paragraph or two about your introduction to your topic. Topic 2 is topic A. Where does this occur? Which ecosystems does this happen in? Which biomes? The whole biosphere? Does it happen in just one particular river? Does it happen just in one particular continent? Okay, so tell me where this happens. How does this happen? So a description of how this particular process happens. And who is affected by this, including who benefits from this particular process. If you talk about air pollution, water pollution, if you talk about pollution problems, someone has to benefit from this process in order for it to be happening. Well, who benefits in air pollution? Well, a lot of air pollution is caused by combustion engines, which includes vehicles. Well, somebody's making a whole heck of a lot of money by us running gasoline through our engines. Boy, we know it on the East Coast right now because of the gas shortage. So who is benefiting from this? Which organisms benefit from this? So include that. So be sure and work really hard on that final project, Milestone 1, and look for my feedback and do the corrections after you get my feedback. My tip of the week for any assignment is to put assignments in the correct format. There are always instructions about this, so make sure you read them because the format is important and it helps me with my grading. It helps me find all of your information. If I have to dig for it, I probably am not going to find what you think I should find. I have seen this time and time again in these assignments, so please use the correct format. Milestone 1, here is the information. You should submit as a Word document or something similar. Double spacing, don't use single spacing. That's very difficult to read, and I don't need any problems with that because I have enough trouble. 12-point font, just something normal. No crazy fonts, no pink font, just use normal fonts. 1-inch margin, I do not measure, but you don't want 3-inch margins either with a little column of text. I've seen it. Submit a minimum of 1 page and a maximum of 2 for the actual information. If you include your references, which of course you should because you need to have 3 for at least every one of these milestones, then that's an extra page that you can add. So there should be 1 or 2 pages of information. All right, this is the format of Milestone 1. This will also be the Milestone 2 format, but when you get to Milestone 3, then you're going to start making the slides, and the final project will be, for example, PowerPoint slides. You don't have to use PowerPoint. You can use Google Slides. I found that out a couple of terms back. And there are other programs that make slides, but you're going to have a presentation like you would be making to a group of people with some slides. So eventually that's what it's going to come down to. Right now it's going to be an outline format, Word document, double-spaced, etc. All right, as always, I hope that you have a wonderful week this week. And if you have any questions at all, m.sigmon@snhu.edu. I am here for you. I can answer anything that you can throw at me, and we'll do it. Also, I want to remind you that every Sunday evening from 6 until 7 Eastern Time, I have office hours online. You can drop in, say something, leave. You can ask me 1,000 questions, anything that you like. So remember that I have that open for you too. Um, like I said, I hope you have a wonderful week, and let's see what you come up with for your final project topics. I cannot wait to see what they are.